Good evening and good afternoon to all our friends in the US, Israel, and wherever each of you connect with us from. My name is Ruby Shamir, and I'm the Executive Director of AIFL in Israel. Today, I'm honored and happy to open the inaugural webinar in cooperation with the Zuckerman Institute. The Zuckerman Institute was established in 2015 to achieve philanthropic vision of Mortimer Zuckerman to enhance collaboration and communication among the most promising scholars in science, technology, engineering, and math in Israel, the United States, and Canada. The Zuckerman STEM Leadership Program, jointly operated by Zuckerman Institute and universities in Israel, the US, enables scientists and scholars to push forward with their boldest research and take the risks necessary to find solutions for tomorrow. The Zuckerman STEM Leadership Program supports future generations of leaders in science, technology, engineering, and math in the United States and Israel. Over time, fosters greater collaboration between the world's most advanced scientific research centers. The Zuckerman Family Program provides vital resources to Israeli universities, allowing them to compete with top North American institutions for the most promising candidates. The program facilitates the return of top Israeli scholars to Israeli institutions, cultivates world-class scientific talent, and in turn attracts outstanding postdoctoral researchers from top Western universities, creating a cycle of excellence. 7 faculty scholarships are given out each year to Bar-Ilan University, Ben-Gurion University of the Negev, Haifa University, Hebrew University of Jerusalem, Technion, Israel Institute of Technology, Tel Aviv University, and the Weizmann Institute of Science. Since the program opened in 2016, 23 Zuckerman labs have been established in Israel. On today's web panel, we will have 2 presenters, a Zuckerman Faculty Scholar from Israel, Dr. Itamar Harel, and his US colleague, Dr. Bernice Benayoun. I'm happy now to turn to my colleague, Wayne Firestone, Executive Director of AIFL in the US, to moderate the panel. Please, Wayne.
Ruby, thanks so much. We're incredibly excited to be launching this series in high science with the Zuckerman Institute, which has made such a tremendous contribution to collaboration and thinking about not just what is sexy today or tomorrow, but really thinking on a much longer time frame about issues that science has to be at the very forefront of. And so we've inaugurated this series. We're so grateful to Lena DeShilton for bringing together this first match of colleagues. You know, a lot of times on our webinars, we bring together panelists from different parts of the United States and Israel who have never met each other. They're sort of talking for the first time. We feel like we're making a shidduch and, sort of introducing some new concepts and relationships. This is not one of those cases. Today, we're actually going to learn from and hear from 2 collaborators that met and have a common interest in a particular fish that we're going to learn about today, but that is only one aspect of their collaboration and their work, but it really gives us a peek behind the curtain. And I think that's a part of what we're aiming at in this series, is to look at really what are some of the critical research areas that science is looking at today that are going to have huge impact, some of them 10 years from now, some of them 50 years from now. But it's important to understand where we are in the process, what we're learning, and frankly, just the relevance to our day-to-day lives that much of this research has. So we can't promise anyone any anti-aging products, that are going to be disclosed on the call today. And we don't need to, because what we really here is to learn and to get a little bit behind the research papers. This is not a symposium. These two are used to doing that. So first of all, we have in California today, up on the West Coast, Berenice Yoon. Dr. Yoon is an assistant professor of gerontology and biological sciences at the Leonard Davis School of Gerontology at University of Southern California, with her proud logo in the background. Her research focuses on leveraging big data methods to uncover new regulatory mechanisms driving the aging process in vertebrate species. And we're going to find out about just how broad a category vertebrate species becomes. So with her I believe in Jerusalem today, Itamar Harel, who got his PhD in developmental biology at the famous Weizmann Institute of Science, and then trained in aging research at Stanford University, where, as you're going to find out, begins some of the collaboration that we're going to explore today. In 2018, he was lured back, and the Zuckerman Institute had a little something to perhaps do with that in naming Itamar a Zuckerman Scholar, and he joined the Department of Genetics at the Hebrew University as an assistant professor. So Itamar, maybe can you, before each of you are going to present today your own research and give us some introductory explanations and an overview of what that is for most of us who are non-scientists. But before we jump into the actual presentations, could you share a little bit about yourself and you know, where this interesting collaboration started?
I just unmute myself. Yeah. So thank you very much for the invitation, and it's a great honor to actually share some of our thoughts and ideas with everyone. So as you mentioned, I currently live in Jerusalem. Before that, I did my postdoctoral training in California, where I met Bernice. We both trained in the same lab during a postdoc. And together we actually, I think we had complementary skills that allowed us on one side, Bernice, to sequence the genome of our favorite organism, and I was able to develop the tools to manipulate this genome, to actually investigate genetics in this fish. For me, it was really a great 4.5, 5 years. I moved there with my family, and it was, I think California was really hard to leave after 5 years. But eventually, you know, our friends and families are here, and we came back to Israel. Yeah.
Great.
Yeah, so, let me take over. Yeah. So, I have like a similar, I want to say, parallel journey on the way to California, where I did my all my studies up to the PhD in France. So I'm from France, and I had every intention of going back after a postdoc. But you know, I spent actually a little bit longer than Itamar, 7 years as a postdoctoral fellow in the Brunei lab, where I actually remember I was there when we interviewed Itamar for the lab, and he did amazing, and everyone was like, "Wow, we really need to like make him an offer for him to join the lab." So it was really organic and natural that after we both, left the postdoctoral positions at Stanford to continue collaborating and try to, continue using these synergies once we established our own lab. So great pleasure, to continue working with Itamar. Great pleasure to be here and talk about it.
That's great. Well, we understand from you and want to explore. Certainly today we have an international audience from Ottawa, Canada, from Kuwait, from New York and New Jersey, go figure, from Palm Beach, Florida. And obviously the topic you all are discovering is one that, touches all of us in our lives in different ways. And we're all experiencing now both the blessing of people living longer and the challenges that are coming with that. And we know that your research sort of looks at really both aspects of that and has implications for that. So why don't we dig in? And Itamar, I know both you and Bernice have not dumbed it down for all of us, but you've made it sort of digestible for us. So why don't we start, Itamar, with your presentation? We'll go through that, and then we'll turn over to Bernice before opening up a bit for questions.
Sounds good. Okay. So again, thank you for the invitation. And if you look beyond, our regular lives, we see, humans around us age quite a similar rate. And in the animal kingdom, we see amazing differences in lifespan. We see over here the bristlecone pine, which is native to California, that lives for several thousands of years, compared to the mayfly over here, a small fly that lives for only a few days. Even if we look at animals which are closer to us, vertebrates, we know that there is 1,000-fold difference between the long-lived Greenland shark on the bottom compared to the very, very short-lived turquoise killifish, which lives for only a few months. So to bring it even closer to home, when we looked at similar-sized fish, the killifish, as I mentioned on the left, lives for only 6 months. The zebrafish, a very, very common genetic model in the lab, lives for 4 years. And the goldfish, maybe not in your houses, it actually should live for about 40 years. So COVID taught us what a logarithmic scale is, sadly. So these guys actually are living on a log scale, meaning that each animal lives 10-fold longer than the other one. These fish are same size, they live in the same temperature, they eat the same food. So how come they evolved such different rates or paces of life? And this is exactly what me and Bernice are looking into, because all these fish, even though one lives for 6 months and the other one for 40 years, they all develop and age in a very, very similar way to ours. They develop cancers, they have reduced cognitive functions, they have reduced fertility, loss of muscle mass, increased frailty. So they all age in a very, very similar way, just at different paces. Looking at humans again, this is a slightly maybe depressing slide, but we actually see here on the right all the different pathologies that we might suffer from towards the end of our lives. You see the graphs, each color. And what we see quite interestingly is the fact that we're very, very protected from many of these pathologies in the first 4 or 5 decades of our lives. But something happened at our 5th decade or around that, and many diseases start to co-appear. And an interesting mind game I like to ask is if we all know how many years we'll actually gain if we will cure cancer, for example, if the entire field of cancer research would actually prevail and we will be able to cure cancer, age-associated cancers, we'll be able to extend human lifespan by only 2-3 years. It is a lot, but some other disease will catch up. So the notion is in the aging field is maybe we do not need to understand and treat each and every disease separately. Maybe another interesting route would be to understand the basis, the biological basis of aging, slow this process down, and maybe delay the onset of all these pathologies at the same time. This way we can actually provide for maybe even up to a decade of additional healthy life for a wide range of people. This is an example of someone that actually did it. Winnie Langley smoked, I think, for about 90 years. And the only reason she stopped smoking was because her vision deteriorated and she started burning her nose while she was lighting her cigarette. So Winnie belongs to a group of people called centenarians, from the word century. They all live above 100 years. And these people share some kind of a genetic secret because they can also pass it to their offspring, to their kids. But all these people actually not necessarily live a healthy life. Some of them, as you can see, smoke, not necessarily, jog that often. But they all have a very healthy aging and almost a compressed period of time at the end of their life, which they start getting like more frail and then eventually die of old age. But they're very, very active. They're very sharp. They work almost to the end of their lives. And this is something that the aging community is very interested in, to figure out what is this genetic combination that allow these people to live such a healthy life. And but I bring you back to the lab now. We can't study, people in the lab. So scientists usually use proxies. And one of the most successful group of organisms which have really revolutionized our ability to study aging are small, very simple animals, such as the Drosophila, a small fly, C. elegans, which is a very small worm, and yeast. These are very, very powerful genetic models that we can, on one hand, manipulate their aging process, and on the other hand, they live naturally short lives, so we can also follow and see what our interventions actually did, if we want to really see if we were able to extend their lives or shorten it. However, because these are fairly simple organisms, if you want to ask these questions using a vertebrate model, we have mice and zebrafish. However, I wrote above them their natural lifespan, which is several years. These several years are the average of a PhD. So we have a problem in actually asking multiple questions and allowing this field to progress rapidly. And this is exactly the reason why we and others have decided to look for a naturally short-lived vertebrate that shares many of the organs and systems with us, but naturally lives for a short period of time. And this is the turquoise killifish, who lives only twice as long to the Drosophila, to this fly, this nice fly that is depicted over here. So this fish lives in seasonal ponds, meaning that they dry every year. Seasonal ponds in Southeast Africa, Zimbabwe and Mozambique. This is me collecting some wild samples in 2015. And as I mentioned, these are the shortest lived vertebrates that can be bred in captivity. There's multiple species, and they actually live in different pace, different rates, so we can compare them. They all display human-like aging and disease, such as cognitive decline, cancer, and what I mentioned earlier. And interestingly, conserved lifespan interventions, such as specific drugs or manipulations of the food, seem to be to work very robustly on this fish as well. But beyond the fact that it's a very nice and colorful fish from Zimbabwe, in order for an animal, an organism of our choice, to actually become a genetic model, we have to have two essential things. We have to have a genome, and we have a genome sequence, and we have to have the ability to manipulate endogenous genes, meaning what biologists do best is, break something and then see what was the effect. We take a gene, we remove it, we add something in, and this is how we actually figure out how it works. Very similar to what kids do in the sandbox. So what we've done is, together with Bernice, this is, of course, where everything was done during our training at the Brunet lab at Stanford, which pioneered this organism, and we were, contributing our role. So Bernice spearheaded the sequencing, the genome sequencing of this fish, and I developed a tool called CRISPR, if Some of you heard about it, CRISPR-Cas9. It's a very useful tool that allows us to very easily manipulate the genome of this fish. So we put the fish together. They actually have a very nice mating dance, and then they lay their eggs underneath the sand. We collect the eggs, introduce CRISPR, and then we can generate genetic models. There were additional papers published on this, on the genome and evolution of this. And the pipeline itself to generate these tools is very, very efficient. We can do it in a few months. For example, we can also introduce new sequences. This is a fish embryo. You see here the eye has beautiful golden eyes. This is the yolk sac. These black dots are melanocytes, just like we have on our skin. And here we generated a transgenic fish that expresses a red fluorescent protein in its heart. So if I turn off the light, I see the heart shining. We grow these fish in recirculating water systems, very similar to tiny aquariums. And this is a course I'm teaching every year in Maine. North of the US, which is a part of an aging course. So overall, if we zoom out, what can we do with this new model? These are the hallmarks of aging, namely the main pathways that are agreed by the aging community to be the most involved in the aging process. And many of those are, the usual suspects, such as damages to our DNA, damages to the ends of our chromosomes called telomeres, damage to our proteins, to our energy powerhouse, the mitochondria. So these pathways were agreed upon because you can either accelerate the aging process by manipulating them or slow it down by genetic or environmental interventions. So what we are doing in the lab is trying to develop approaches to use this fish and model processes which are currently not possible to model using mice or zebrafish or the non-vertebrate models. We are focusing, for example, on genes which were invented, quote unquote, only within the group of vertebrates, and also model specific diseases and provide a platform to test new drugs and maybe to better understand some of the diseases that affect all of us with age. So just to conclude, what we're doing in the lab is trying to figure out how a disease works by generating disease models. We're also very, very curious to try to figure out what are the different molecular pathways which actually drive an organ-specific aging process, because we tend to think about the aging process as uniform. But as you saw, and then you probably know from our own lives, some people are affected by neurodegeneration first, which is our nervous system, our brain. Some people have, immune system problems with age, and other organs are affected at different rates. So there is probably a different mechanism that governs each and every organ, and of course, they probably share some pathways as well. But this is a very big focus in the lab. And also, quite recently, we tried looking into how an individual cell ages, because we have multiple types of different cells in our body, and each of them supports a different requirement. A heart cell has to pump and contract all its life. A neuron has to survive throughout our lives and perform complicated tasks. So this thing actually probably gives rise to different signatures of this process. And we're very curious in using this fish in order to also follow the aging process from outside without intervening. Meaning, you remember the original fish that I told you about in the first slide with the red heart? So this is how it looks like when it's an adult. We can actually see the heart through its body. So imagine about, following the aging process from outside an organism and trying to figure out when aging actually begins. So this is these are the questions that we're looking into in the lab. And I just want to have a spotlight on the people that are actually doing most of this work. This is my fantastic lab. We started our lab about 3 years ago. in the Zuckerman Institute at Givat Ram, which is a beautiful campus just next to the Botanical Gardens of Jerusalem. These are my funding sources, and I'll think I'll now, give Bernice the stage, right?
Well, that's great. Unless you want to give a pop quiz to the audience to see. No, actually, Itamar, that was perfect and really a fascinating introduction to the topic. So let's continue with Bernice, and then we'll start hitting some of the questions afterwards.
Sure.
Sorry, I was muted. I'm very happy to try to talk a little bit more about what we're doing and how that integrates with our collaboration. So I started my lab in the summer of 2017 at the University of Southern California in LA. And the big goal we have is to understand the regulation of vertebrate aging. And so, if you remember one of Itamar's slide where he showed, there's like those non-vertebrate organisms, which really have been the powerhouses of the field, moving the field of aging research forward because they're so easy to work with and so short-lived, but they have some limitations in understanding things that are specific, right, to vertebrates like bones and blood and an advanced immune system. And all of these things we know participate in the experience of aging in humans. And so in my lab, we do work with the fish, but we have more generally an interest in vertebrate models. We do use the laboratory mouse because it has a lot of tools that are very well developed and I can say that as the person who assembled the killifish genome, the killifish genome is nowhere near as well assembled as the mouse genome. It's just a matter of technology and investment, right, to get there. So the mouse is really powerful as a discovery tool, but it lives forever in lab terms. And so the killifish really brings us to the point where we can very easily and quickly test important hypotheses on the aging process. Obviously, the goal of my lab's research is not to make cute fish and mice live long, healthy lives, but humans. So when we can, we try to integrate data from human samples, although it's always more technically challenging for obvious reasons. And so with all of this, we're interested in a number of compartments. And I'm just briefly going to say those three cell types, macrophage, monocyte, neutrophils, they're just immune cell types. And the reason we're very interested in those cells is because it's not pretty clear that immune dysfunction is one of the major things that could drive a lot of aspects of aging through notably inflammation, which is a word that you guys probably heard. This is why you take like Tylenol is to decrease inflammation or things like this, but it's known that inflammation increases with age and might actually have damaging effect on the rest of the body. In terms of tissues where we can, for instance, look at this, we look at the brain because obviously we're all very committed to, slowing down the aging of the brain. That's what's most scary, I guess, to most people. And then because my lab's really interested in sex differences, and I'll come back to it, we're also trying to understand ovarian aging and menopause and the impact it has on systemic health, because it has a lot of impact on women's health. With all of this, we use that big data approach that Wayne mentioned in his intro, where we just look at everything we can in terms of all the molecules in the cells and the tissues and see how they're modified with aging. And then sometimes also how these modifications might be prevented in response to pro-longevity interventions, while also checking how is that different between females and males. And the idea is if we can figure out exactly what goes wrong at the molecular level, we can then go back to the cell or the tissue level and try to fix it and see if we fix the functional problems that we know occur with age. Can we fix defective immune response? Can we fix defective, cognition? And then, of course, the pie in the sky, what we really want to do is then go all the way back to the organism, the fish, the mouse, and see if we can actually improve aging in a whole organism. And that's really what we're trying to do in my lab at USC. And so I wanted to talk a little bit about why we're so interested in sex differences. You may or may not know that the majority of studies that are done in biomedical science are usually done on only one sex of animal, generally overwhelmingly males. And there's a lot of reasons that are being brought forward by people who do this, like, studying both sexes is too expensive. Female data is so noisy. Females are just males with hormones and all sorts of things that are not really reflective of real biology. We actually know that in humans, women outlive men by about 10 years on average. So it means there is something fundamentally different. But again, because of that bias in research, there's very little that's known about the differences in the process of aging in males versus females. And so again, this is really important in every realm of biomedical science, but it's, I want to say, even more important in the aging, because we know, again, like human, huge sex differences in lifespan, but also, there's now 15 years worth of work on interventions to promote longevity in lab animals. And the biggest thing we've found in a couple of things that work in lab animals, but the biggest thing we've learned is that nothing works the same in males and females. And generally, it either works only in one sex or works much better in one sex than the other. And what that tells me as a scientist is that if the aging process was exactly the same in females and males, then the exact same intervention should have the exact same effect on aging in both sexes, and it doesn't. So it means this is something we need to understand. We know that the way genes are regulated in female and male cells is very different. And that's not just in reproductive organs. The liver is actually one of the most different organs between females and males in terms of the ability to process drugs, take down drugs, so drug metabolism and things like this, very different. And then we know that age-related disease like Alzheimer's, osteoporosis, cardiovascular disease, all of those skyrocket after menopause. Actually, the best predictor of a long lifespan and a healthy lifespan in women is a late age at menopause. And so the idea here, you could be like, well, okay, we're only going to be benefiting women if we look at this, since we know like they're living longer and everything. But really the idea is if we understand those fundamental processes, we could actually use them to improve both male and female lifespan in the end. I mentioned we're very interested in immune cells and inflammation. And that's because, and this is sort of a little cartoon from a review on the topic, we know that for some reason, every time you experience a wound or an infection, your inflammation levels are going to increase. And they will go back close to where they were before, but never quite where they were before. And it looks like with aging, the accumulation over a lifespan of not going back, to baseline leads to this inflammatory milieu, which promotes tissue dysfunction. And so it's a really big problem. But there's a lot of hypotheses as to what drives this, but there's not much understood. And so really what we are trying to understand with that angle of really always trying to understand what it might be different and how it's differentially regulated in females and males is how inflammation might be driving vertebrate aging. And so we're interested in mechanisms that could drive that inflammation with age. And so, as Itamar mentioned, the killifish is actually a great model to study at least one aspect of this. Now that we have all of this pipeline, right, the genome, the annotation of the genome, the ability to modify the genome, we can now study this more in depth. And so specifically, we are investigating, and I just want to highlight the fantastic people in the lab who are doing the work. We're trying to understand how transposable elements, and I'm going to explain what they are, might be driving vertebrate aging, notably by promoting sort of a constant state of immune activity. So transposable elements are a huge component of our genomes. If some of you were around in the early 2000s when the human genome sequence was first published, I'm sure you heard the term junk DNA, like we have a lot of junk DNA. It even made it in pop culture and TV shows. And the idea is that they were seeing those transposable elements in the genome and they were like, well, that serves no purpose. So it's junk, it does nothing. And that has been largely debunked. We actually think, thanks to a lot of work, notably from the ENCODE project, that most of the genome accomplishes some function. It turns out that those transposable elements are sort of relics of infections past, not infections of us, but of our like great ancestors throughout evolution. They're basically genetic elements that are derived from ancient viruses that make up a huge portion of our genomes. And so for the human genome, 45% of the genome, so you can see the pie chart here, is actually constituted of transposable elements. And for the killifish genome, which makes it a great model to study the impact of this, it's estimated to be at about 60%. Those, because they come from ancient viruses, they are basically indistinguishable to our immune system to viruses. So when they get active and they start moving around, and which is why they're called jumping genes, you can actually get an immune response to them. And what's really interesting is that there's now an accumulating body of work, including some work that we did, like when I was at the end of my postdoc, showing that they do reactivate with age. So normally when you're young and healthy, there's almost no activity of them, but they get activated with age. They start being there, present and detectable by the immune system. And they're also upregulated in tissues where age-related disease occurs, like Alzheimer's or cancer. And because they can drive that immune response, and also because when they jump in the genome, they can break the DNA and damage it, we actually think that activation of transposable elements may drive aging phenotype. So we're really curious of using the killifish and modifying the ability of the fish to suppress or release suppression on those transposable elements. And that's the project we're actually collaborating with Itamar on, to understand the impact of this on aging. And so this is just one data slide that I wanted to show, and hopefully I can explain it simply, where we've measured the de-repression of those transposable elements across 4 tissues with aging in both female and male killifish. And so the color code is basically, if it's blue, it's male. If it's pink, it's female. Sorry for the gender normative color scheme. And if it's darker, it's the old animals. And if it's lighter color, it's the young animals. And you can see in all the tissues we've looked at, there is a general upregulation. So if it's red, it means it's higher. If it's blue, it means it's lower. And so you can see there's a higher amount of those transposons in all the tissues with age. And we see this both in the males and the females. There's some differences in what's going on between sexes, but I'm not going to go into the details now. But really that suggests that we're going to have a great model to understand how de-repression of those transposons might drive aging. And maybe why not understand why the killifish is so short-lived, because they actually start activating those transposons much earlier than a lot of other vertebrate species. And so really, just to conclude on the killifish again, to like show you why it's a cool model and why Itamar and I are so excited to work with it and develop it, is that you can recapitulate an entire human lifespan in 4 to 6 months in the lab. And with all the tools we have now, we can do modern genomic big data analyses to understand important biological processes. Thanks to Itamar, we have the ability to modify the genome in this fish extremely fast compared to other species. Because there's also genetic variation. You saw Itamar getting some wild samples from that puddle in Zimbabwe. Those fish that are captured in the wild have various lifespan. And so we've been able to actually look at how those differences between fish from different puddles might relate to different rates of aging. And then we can also compare the killifish to, like, for instance, the zebrafish, much longer lived, and see, okay, what's different? How did that species evolve such a short lifespan? And so I hope that was clear enough. And like Itamar, I think it's always important to explain, when you're a lab head, that you don't do the work yourself. Most of the work is done by fantastic students and young scientists. And so I really want to give it to them, like, none of this would be possible without them. None of it would be possible without the sources of funding and or the collaborators, including Itamar. So I thank you all for your attention as well.
Wow. I mean, what the two of you have shared with us today in really, frankly, such a clear and concise way, we already have tons of questions, so I'll try to get to as many as I can here. Not surprisingly, a lot of people, as soon as you sort of mention this, the archaeological role that you're playing in looking at the genome, and looking at ancient, essentially viruses that you can still learn from. The immediate question, since this webinar is happening in the middle of a pandemic and science is on this incredible race, what have you learned? I want to come back to the specifics of your research, but people are really, I hate to use the term dying to know, but people want to know what has accelerated and what have you learned that may actually have relevance to some of the pharmaceutical work that's being done now looking at the COVID virus.
So I wouldn't say that the transposon teaches something about COVID, because the level of virulence is very different, right? We would not be able to survive if those ancient viruses in our genomes were as active and as virulent as COVID-19. We do have some research in the lab. And again, I want to highlight that both Itamar and I do like preclinical research. So, I don't want anyone to do anything, to themselves based on what we say. But I can say some of the work we've been doing on the immune cells I mentioned could provide maybe some ideas with giant caveats of why COVID-19 is worse in men than in women. Because we've noticed that the male immune cells tend to overactivate upon challenge more than the female immune cells. But again, I would really leave the clinical considerations to the clinicians. I don't want to mislead anyone on anything, on the importance of what we're doing, which is still important, but not necessarily relevant to COVID.
Another thing I can add about this is the fact that COVID-19, as among many other diseases, is actually an age-associated pathology, because we see that older people are way more susceptible than younger. And this is exactly one of the reasons that we're trying to figure out how the aging process actually manipulates our biology. So I think I can add COVID as one of the other pathologies on my slide, because again, if we are able to manipulate the aging process, we'll be able to be more resilient to outside infections, because COVID-19, a lot of the things that basically are making people more susceptible is too of a strong response. It's an overshoot of our immune system, which maybe by tuning it better could have been a much simpler or less harmful response for the body.
Interesting. So let's start with some of the basics. When you chose the killifish, did you get lucky and just choose the right one? Because it sounds like it's provided you with great...
No. This fish was collected in 1968 for the first time. And since then, it has been very, very heavily characterized by multiple groups around the world. And they basically characterized its aging process, how the brain ages, the muscle ages, you have a bit more cancer. So this organism was very nicely characterized, but it didn't have any genetic tools. So what we added, Bernice and I, were the tools to transform it from it's
like a cool fish that looks pretty from like a useful lab animal, basically.
Yeah. So it was there. It was collected. There's actually, if you Google it, these fish are beautiful, and there's collectors of killifish almost, I think, in every continent you can look for. Even in Israel and in Germany and the US, there's multiple ones. So a lot of people are really following their biology and their breeding. So we had a lot of information to base our studies on.
And I know, Bernice, you mentioned also the zebrafish. So are there others that have been identified that other groups perhaps in around the world are working on in sort of similar fashion?
For you mean different fish species?
Yes.
Yeah, I mean, so the zebrafish is really like the big one. I would say some people are studying, I believe it's the cavefish, because of its adaptation to lack of light, right?
Yep.
And so that allows us to understand vision and pigmentation and things like this. Really, we might sometimes oversimplify why we use a specific model, but really usually in the lab, when we choose to use a specific model, like be it the mouse, the killifish, the zebrafish, we usually use a model for its specific biology that we can study to learn something interesting, right? So A super short-lived organism is a great organism to learn about aging, and so is a very long-lived organism, right? The Greenland shark or the naked mole rats, which are the same size than a mouse, but they live 30 years, right? And so this is really what we do as biologists. We try to find the organism that is the best suited for whatever question we are trying to answer.
Got it. So somebody just put a question in that is not intuitive. How do you measure cognitive aging in a fish?
This is actually quite simple. You can do it if you have an aquarium at home. You can do that. You can see it yourself because when you at some point, I have an aquarium and in my house, and at some point, you come towards the aquarium and the fish swim up. They know they can anticipate the fact that they can be fed. So what you do in the lab is you train the fish. You come every morning, you shine a red light, and you provide food. And then the fish learn to couple between these two events, the red light and the food. And if you only shine a red light without giving the food, they will actually swim up because they will anticipate the food coming. And
it's basically the same idea as Pavlov's dog, right? It's
exactly. Yeah, conditioning. And then if you teach an young fish these tricks, or you teach, an old dog or an old fish these new tricks, you can actually measure and see that the old fish don't learn as well. And this is a very powerful tool because think about, we Bernice and I will identify a mechanism that can slow the aging process. We can use these tests to evaluate whether the physiology of this organism is actually better now. Can it learn towards even a later age of its natural life?
So there's what could potentially be a provocative question here, but I'd be interested.
Go ahead.
Is aging a disease?
That's complicated. If you ask the FDA or its European equivalent, it's not. And that's why actually we cannot say that there's any clinical trials, even though we know in the lab of a number of things that work in delaying aging in lab animals. None of those can be applied to humans because aging is not considered to be a disease by the FDA and its European equivalent, which actually makes it kind of frustrating, right? Because everyone's always asking you when you work on aging, oh, why can't I do it? And it's like, well, I wish I could give you something, but it's not legal. So the one thing that I would say is it's not necessarily a disease as in it's not abnormal, because pretty much if you live long enough, you will get old. That's like one of the big truths of the field. But we think it underlies all of the so-called age-related diseases. And so there's some smart people, notably Nir Barzilai. I don't know if you've ever had him on your platform, but he's in New York, and he's trying to use the fact that we think those age-related diseases are symptoms of aging to test whether things we know work in animals would work in humans. So there's a trial, the TAME trial, that is currently ongoing, where they're giving metformin, which is an anti-diabetes drug which has anti-aging properties in lab animals, and they're giving it to people who already have one age-related disease. And the idea is to look at the interval to the next one, because the idea is once you get one, usually you're going to get the second one within 3 to 4 years. Like if you have diabetes, you're going to get high blood pressure or whatever, right? And so, and the idea is if it works in humans, we should be able to see a lengthening of that interval between disease diagnosis. And so that's ongoing. And if that works, I think that's going to be really powerful. But yeah, people are trying to see if we can treat it the way we would treat disease, but instead of treating the symptom, which is really what we do when we treat disease, try to treat the root cause.
Mm-hmm.
Okay. Are there already examples? You made a reference earlier, Bernice, to interventions. Are there interventions that are known to work now, at least in animals, that are promising?
I mean,
so
go ahead, Tamar.
Yeah, something short, because the thing is that I don't want to give this notion that we have these secrets and we're not sharing because we can't. The thing is that overall, all our interventions and all our knowledge so far, I think that what we know is conserved all the way throughout all organisms is actually things which almost are a good advice from our grandmother, which is basically eat a balanced diet, have a good emotional connectivity with your environment.
Exercise.
Yeah, exercise, don't be stressed. And all these together will or are shown to extend your lifespan by an average of like 10 years. And this could be way, way beyond any drug that we will identify. So the secrets are within our grasps, even though it might be simpler to go to CVS and get prescription. But we do know there's hotspots.
One of the most important things that, Itamar's mentioning is very conserved is caloric input. Calorie restriction or dietary restriction in general is the one thing we know works from like yeast. It extends lifespan of yeast. It extends lifespan of worms. It extends lifespan of fish. It extends lifespan of mice. It's controversial as to whether it does it in monkeys, but there's good data that at least it improves the health of monkeys. And there has been some trials in voluntary humans, right? Nobody was forced to do that to themselves. And it does look like it improves health. The problem is that it's not necessarily feasible for humans to be on, 40% reduction of caloric intake. And when I say 40% reduction is not, you were eating a lot of chips and you just cut that out. No, it's 40% reduction from what you would normally get if you were eating optimally without losing vitamin input. And you need to be followed, by doctors and everything. But it looks like that's the crux, right? It's really nutrient input. Caloric input seems to be the big crux of the regulation of aging rates within a species.
You just addressed Jacob's question head on, so thanks for that on the caloric research. Let me. I want to go back to that chart that you showed early on, Itamar, showing the those of us in that post-50s range, right. Sort of making sure we go for our annual checkups and eating lots of greens. And killifish. Are we allowed to eat killifish, by the way?
If we're allowed to, I don't know. I don't think there's any laws on which fish you're allowed to eat or not, but you can.
Fair enough. My question is about like if your work shows that actually people can live longer, and we learn some things, pay attention to women's bodies and research in women in female animals, as Bernice shared that fascinating insight about perhaps where bias in research is really short-sighted for all of humanity. Yeah. My question is, because we see that if people aren't healthy as they live longer, and if they develop, particularly in the brain, we know some of the morbidity you were showing on the slides. How are you able to sort of address both of those things in lockstep so that we live longer potentially, but we live longer not with more disease or as we're alluding to?
This is basically, I mean, if you think about human lifespan 100 years ago, it was much shorter. And by developing antibiotics and vaccinations and better hygiene and proper nutrition, human lifespan has dramatically increased. And your question is very, very on the point, because, the nightmare scenario in the aging field is extending lifespan, but not extending our health. Right? Because I would be very, very happy if we won't even extend lifespan at all. We'll just, compress the period which we're sicker towards the end and increase the proportion of time we spend healthy. And a lot of aging studies are doing exactly this. They're not interested in extending the life of the organism, just pushing the onset of pathologies later and later and allowing for longer health. So
that's actually been popularized in the field as the concept of health span.
Yes, exactly.
Lifespan.
Yeah. So I don't think, I mean, again, me and Bernice are working on the basic science, but if it will be interpreted by others for application, I would really be in favor of promoting health span and extending health span and allowing us to live our lives just with, more active and better health instead of promoting a longer life.
So I'm going to try to get a couple of short more questions in the.
Sure.
Are you able to measure the effect of growth hormones on immune responses?
So growth hormones and do you want to take this, Bernice?
Yeah, I mean, we don't necessarily work on it. Growth hormone is actually kind of a controversial thing because I know there's a lot of clinics here in LA that offer growth hormone treatment as an anti-aging thing. But if you ask people who work in aging biology, this actually makes us kind of chuckle because high growth hormone signaling is shown to accelerate aging. So we haven't necessarily looked at the immune compartment, but I can tell you one of the biggest life extension in mice is when you cut growth hormone signaling.
Yep. And there's actually a pathology. There's a genetic disease in humans which causes dwarfism, and this is one of the mutations is growth hormones, and these group of people actually suffer less from some of the age-associated diseases that
specifically affect cancer. They have a lot less cancer. Yeah,
so it is implicated also by chance because there's a pathology in humans, but reduced growth hormone is actually associated also in humans with a reduction in age-associated diseases.
So, specific question about the BRCA1 and 2 genes, and the research that we know now about breast cancer. Bernice, is there work in that area? Are there interventions already in that area that have shown any promise?
So I don't know that there's any direct work in the aging field on the BRCA genes. The BRCA genes are really something that the cancer field's working on. Obviously, gene editing techniques like CRISPR are a big hope for people who have these kind of mutations, but I don't know if it's quite ready for the clinic yet. But DNA damage in general, and the BRCA genes are actually encode for enzymes that repair DNA damage. DNA damage and DNA damage repair in general, so not necessarily BRCA, is something that there's a lot of people in the aging field who are interested in looking at.
And actually, it links quite nicely to the transposons, because transposons actually emit a signal as if there is DNA damage, and that's how they activate the immune system. So there is a link between these topics.
And can you actually target transposons for specific ailments or
so don't forget that transposons are very nicely shut down when we're young. So the mechanisms that keep them at bay are working very well throughout our lives. They're just loosened a bit when we age. So the idea is to better understand how it works naturally in our body when we're young, and just recapitulate it or phenocopy it when we are older to push it towards. So I think the probably Bernice would agree with me that the hope is that we'll find a druggable target that can put these guys at bay without any need to edit them, because they're so numerous we won't be able to.
It's basically, just to give you an example, line 1, which is the most frequent transposon in the human genome, has 500,000 copies, independent copies in the human genome. So you definitely don't want to have to target each of them separately. But there are some sort of tricks. The Sedivy lab has shown that you can use some of the drugs that are used for HIV patients to shut down some of the transposons in older animals. The problem is, I just want to again put a huge caveat. That doesn't mean people should jump on anti-HIV drugs, because they have a lot of very unpleasant side effects. But that means that in theory, if we can control side effects, it is a druggable target.
Wow. Well, we're actually coming up at the end of our hour, and I didn't get to all the questions, which is just really, I think, just a testament to the fact that your work is really so important and on the minds of so many people today. And so I'm going to lead you. I'm going to end with a question for each of you, just to maybe a brief answer on. You started this professional collaboration in your postgrad work. What would this conversation be like 10 years from now? Like what do you anticipate the kind of issues and the kind of progress at the rate at which you're seeing now? Like what is possible 10 years from now, 20 years from now? What's the trajectory that you see science in your field headed, and what are going to be some of the defining issues, for maybe not, for our children and our grandchildren in years to come?
Yeah.
So, do you want to take it first, Dimash?
Sure. So I don't know, citing X-Men. Every few generations, science has a leap. No, but I mean, in a way, it's hard to.
X-Men fan, that works.
Yeah, me too. Yeah. So I mean, in a way, it's really hard to predict, because I'm predicting based on the tools that we have now. But just like vaccinations revolutionized health for human species forever. There's something around the corner that might arise that will change our health forever again. But if you would ask me what I would predict, I would say that I think slowing down the aging process using pharmacological interventions will be something that will be used, I think, quite broadly in order to treat age-associated diseases and slow them down. I think this approach will eventually develop, and just like we use antibiotics for infections, we will use these interventions to slow down dementia, slow down cardiovascular diseases, and so on. This will be a very, very important way to, not a silver bullet, but it will be a very important way to treat these pathologies.
Yeah, and there's like Itamar says, it's hard to predict because there could be like a breakthrough tomorrow that would change, the current course of things. But there is a number of technologies that are, being tested, or hypotheses that are being tested in animals that are very promising. And again, like with the normal pace of research, going from preclinical to clinical, I do believe that we're going to get a reduction of those morbidities, right? Those comorbidities that occur with age within the next 10, 20 years. I'm pretty sure we will see that happen. Mm-hmm. Extension of lifespan itself, I am less sure we'll see it in the near or even midterm future.
Yeah. Well, and I'm not sure we should.
Yeah.
No, a point well taken. And so much, you've given us such a rich understanding of your respective research and collaboration here. And there's just so much you've opened us up to. And really, frankly, that is the purpose of this series. And we're already seeing many people responding, asking for more of this. And I think that is a recognition that we can't put science just out there. It needs to be part of the public discussion. It needs to be part of the prioritizations that we're making. And in order to do that, we have to become more literate. We simply can't say, oh, that's something for people in laboratories to worry about. We know, and if anything, and I don't think there's a silver lining to COVID at all, but we recognize that science has created our own form of miracle in creating a vaccine in such a short period of time. It's really a tribute to what scientists are capable of doing and what you have been trained to do, and nobody can do it on their own. So collaborations like this that the Zuckerman Institute is supporting are so fundamental to the kind of what I would call infrastructure of the future that you all are a part of. We are grateful for these insights. We're grateful for your participation today, and we look forward with our audience to explore many topics like this in other fields of scientific collaboration that are going to be important for all of our futures. We do these webinars. We've been doing them twice a week, Wednesdays and Sundays. religiously, if I could say, since we began COVID. We're over 100 episodes in now. We continue this coming weekend looking at how the Israeli and American educational systems have what they've learned over the past year, the challenges and the learning opportunities in school systems, K through 12. We have some outstanding educators and administrators for that program. We invite you back, and we hope everyone has a safe week and will join us for future science episode series and for all of our webinars here at the American Israel Friendship League. Thanks again.
Thank you.
Thank you so much.